Hello, and welcome back to part 4 of our series of mini lectures on the respiratory system. And what we're going to look at in part 4 is some additional characteristics associated with the respiratory system. Now, what we talked about in the previous lecture focused in on the exchange portion of the respiratory system were the type 1 alveolar sites, so the very flat, simple squamous cells that form the barrier for the air passageway, as well as the type 2 alveolar sites, which are producing surfactant that kind of coat the type 1 alveolar sites and minimize surface tension and make sure that the airways remain patent, they remain open and pliable. There are also going to be some other cells that we're going to see, and the other cells are going to be the alveolar macrophages. And the alveolar macrophage, in some books, may also be called the alveolar phagocyte or the dust cell. And these are similar to macrophages that are found throughout the body. They're all derived from monocytes. And they're going to be doing things similar to what macrophages do. And so you can find these macrophages crawling through the connective tissue within the alveolar septa, the alveolar walls, or you can find them actually on the alveolar surface themselves. So they essentially crawl out along the airspace and essentially clean up debris which may be present there, or clean up damaged regions as it's going through. If we take a look at this, it becomes very prominent. We got electron micrographs of alveolar macrophages on the left side from a non-smoker, on the right side from an individual with a history of cigarette smoking. And you can see the presence of lysosomes in both cases, but and the alveolar macrophage on the right-hand side, you can see large residual bodies where essentially the lysosomes have been used to try to break down these materials, but there's an accumulation of this essentially particulate matter that's been cleaned up from the inner surface of the lungs. So the macrophages are going through trying to clean it up, minimize the effect that this would have on the diffusion boundary across the surface, and eliminate the things that could cause potential damage to the lining cells. But you can see that there's a pretty dramatic difference between these two cells. Now, if we take a look at kind of the three-dimensional structure of the exchange portion, take a look at the alveoli, we actually see that there are structures called alveolar pores or pores of Kohn. And these are essentially openings that go between one alveoli to the next alveoli. And so they're kind of like little shortcuts. So they're not in the main passageways, the main airways, where we've gone from essentially the alveolar ducts down into the alveoli sacs. These are essentially going from one to the next, so essentially penetrating through the wall. Now, these are important if you have damage to a region of the lungs because it allows for collateral exchange of air. So it essentially, if you've got a passageway that's been blocked, you can allow air to flow through these alveolar pores and allow for respiration for exchange to occur. So it's good, but it also has a possibility to essentially be a passageway for macrophages, again, which is good, but it also can cause for the spreading of disease, either neoplasms or pneumonia. And so we can take a look at this. They have a role in a good, healthy lung, but they can be obstructed in chronic lung disease, or they can be a mechanism for the spread of disease in things like pneumonia. Now, if we take a look at the lung, we need to recognize that it has a dual blood supply. The major supply is going to be coming from the pulmonary artery, and this again is going to be going down into the lungs all the way down to the exchange portion where we're getting into the actual capillaries that are going to be throughout the alveoli and the alveolar sacs, which are going to allow you to have the exchange occur. Keep in mind that the pulmonary artery, though, the major supply is deoxygenated blood. It's blood that's coming in with the sole purpose of getting rid of waste materials and drawing in oxygen. We're also going to have a bronchial artery, a minor supply that's going to be coming in. This is a branch off of the aorta, which is going to be coming into the lungs, but it's going to be carrying in oxygenated blood. And again, that's important because we need all of the cells within the lungs to remain alive. So they need to have a good supply of oxygen and nutrients, but primarily oxygen at this point, so that they can do cellular respiration, so they can produce ATP, so that they can survive. If they were dependent upon just the pulmonary artery just receiving the deoxygenated blood, it could impair their function. And so we've got a dual blood supply coming to the lungs. Both of these vessels are going to be entering through the hilus. They're going to be following the respiratory passageways down into the deeper portions, the exchange portions of the lungs. If we look at the outer surface, we're going to have the pleural cavity. So we're going to have a visceral pleura, which is the outer surface, the outer lining of the lungs, and the parietal pleura, which is going to be the inner lining, the lining of the thoracic cavity. Both of these are going to be a mesothelium. So we're going to have a nice smooth surface. We're going to have a serous fluid, a proteinaceous fluid, which is going to be produced between the parietal pleura and the visceral pleura. And so we essentially have a smooth, moist surface so that the lungs are able to move freely from one another and freely from the thoracic cavity lining. You don't want essentially the lungs to become attached to one another or attached to thoracic lining, because you run the risk of impairing the movement of the lungs, impairing the ability to breathe in and breathe out. Now, taking a look at a couple of disease processes, emphysema is an example of a progressive disease of the lungs. You're essentially looking at irritation, which is going to be occurring deep within the lungs. It's going to cause macrophages. It's going to cause neutrophils. They're going to go through. They're going to try to clear up whatever is causing the irritation, the particulate matter, generally cigarette, nicotine, smoke, things like that. The neutrophils are going to be releasing things like elastases, which are going to break down the elastic fibers. The macrophage is going to be going in, cleaning up the area. We're going to have a buildup of scar tissue. And what you end up with is a breakdown of the elastic fibers, a buildup of scar tissue. And so you end up with less pliable lungs. And so they're not as good at expanding and recoiling. And so it makes it harder to inflate the lungs and harder to ventilate it, and basically harder for exchange of air in and out of the lungs, which impairs the ability of diffusion of materials into the bloodstream. In asthma, what we're looking at is essentially an allergic response. We get some type of allergen, which is going to cause mast cells to release histamine, like we've talked about before. The histamine can cause a leakiness within that area, so we get tissue fluids that build up, we cause edema to build up. As part of the response, we can also see constriction of the smooth muscle. We talked about the smooth muscle within the walls of the bronchioles, the smaller bronchial structures. Constriction of the smooth muscle cells cause bronchial constriction, but essentially it's going to cause enough constriction that it inhibits airflow to the deeper areas of the lungs. So it makes it, again, difficult to exchange air between the respiratory portion of the lungs, the exchange portion of the lungs, and the outside world. And so it makes it harder for oxygen to be delivered to the deeper regions, to the lungs, harder for it to get into the bloodstream. It can also cause a hypersecretion of mucus by the goblet cells, and that mucus then will kind of glom up, kind of help block the passageway and inhibit the exchange occurring as well. Cystic fibrosis is another disorder associated with the respiratory system. Cystic fibrosis is associated with a normal protein which is involved with the pumping of chloride along the epithelial lining. Normally water would follow the chloride and dilute the mucus, and then by diluting the mucus we could prevent the buildup. In patients with cystic fibrosis, the protein that's normally pumping the chloride across the epithelial lining is not working properly, and so because chloride isn't pumped across the epithelial lining, water isn't following it, and so what happens is mucus isn't diluted, and mucus tends to thicken and accumulate. And so by thickening and accumulate, it kind of blocks off air passageways. It serves as a location where bacteria can become established, and so interferes with the functioning of the respiratory system. Um, and then the last two slides are essentially looking at key features of the respiratory system, and these are in the handout. They help you by identifying where you are within the respiratory system by looking at the combination of the epithelial lining and the support structures within the walls of these air passageways. And again, that helps you then to identify where you are and identify what's going on within the respiratory tract. This finishes up our discussion of the respiratory system. The next series of lectures is going to be on the circulatory system, again, tightly associated with the respiratory system as well as other systems within the body. If you have any questions, feel free to email me at hoffmanj@arcadia.edu. Hopefully you come back for the circulatory system lecture.